currently going to overlap the nucleotides. So I'm going to turn that off. I come up here to view and turn off the zoom stripe label. Now when I zoom in, let's zoom in right about here. You can see all of the nucleotides, again, as the individual letters. You can once again, within the gene model, see the amino acid translation. And now when we zoom in on the read, what you can see is only the letters where it does not match the genomic sequence. As a quick note, one of the things that you can do, aside from adjusting the color of the tracks and adjusting the color of the background, you can either come up here to File, Preferences, or use the one-click button from the toolbar, and you get to the Preferences window. And this covers all sorts of preferences within IGB. But if you come to Other Options, you can adjust the residue colors if you're looking for instance, specific mismatches and wanted to change a color. You can also find the code on display if you want to hide it, or if you prefer a 3-letter amino acid representation. I'm just going to leave everybody the way they are. Now, if for some reason you wanted to specifically restore the read sequence, right here, I clicked on the read to highlight it, and then I use a right click, or if you're on Mac, a control click, to access the specific menu. And if I restore read sequence, what you can see is that for that read, the sequence is completely back. And then I can switch it to mismatches again. So sequence is always available, it's just not always needed. Now, while we're here, if you notice, I am in the Advanced Search tab. One of the really nice features that we put in is the ability to search for residues. This is an exact match search. This is not BLAST. The program will not do an alignment for you, but it will search out for you any sort of sequence that you ask it to. So the first thing I'm going to show you is how we tend to use and map our primers for PCR. So here's, once again, our gene, and you can see right here, there's an alternative exon that's sometimes retained and sometimes spliced out. So we created primers so that we could pick this out. So my first primer is going to map into the alternative exon. Now notice I'm set to residues. I'm going to click on overlay results. And what overlay results will be is that it will draw for me the very first mark right there. If I double click it, I can zoom in, or I can even double click the highlight itself. And this is the exact sequence I asked for. So now I want to map the primer that picks up the splice site. But again, I need to point out that IGB uses an exact match. So since this primer actually overlaps the intron-exon boundary, I put a pipe in it. If I were to try to search for this sequence, it would fail. See, it found nothing because this sequence is split at this point. However, if I map the left side, which popped up right there, and then the right side, I now have, again, mapped to the model where my primers are. And then finally, I put in the common. And by using the sequence search overlay, I can actually map out all of the primers that are going to be part of identifying this area. And it's a great way to keep a record of it. It's also fairly useful if you have data that's loaded to know what your primers are going to pick up in your samples. So there's another time we really like to use this overlay feature, and I'm going to zoom out a little more. And what we do is we can map promoter elements. So I'm going to clear this view by using the X, and it clears everything out. We're still on the residue search, and I'm still going to overlay my results. Here, what we're going to look for is a heat shock element. And what you can see is that the IGB terminology uses a period as our wildcard. If you're interested in seeing some of the other ways to query, for instance, if you know it'll either be a C or G, you can use these brackets. This information, again, is in the user's guide and is in advanced search. All the other ways of using the advanced search are here. And like I said, I'm just going to point out one or two other ways to do this search. So here I've entered it with the dots, and right there is the heat shock element. I know that I have several Tata boxes that I would also like to see, and you can see they popped up right here. And I'm going to zoom in for just a moment because I'd like to point out that IGB will show you whether this matches to the 5' or both sides or just the 3'. AT is going to obviously be the same in both directions, but here you can see that there's very intentionally a gap indicating that this maps to the 5' side. So we will also look for a morning element. And what the morning element is it's a binding site for genes that are part of the circadian pathway. And again, both of these are on the 5' strand. You can see by the gap below. And just to show you what it would look like, although I know that this isn't in our gene, this right here indicates that my core element will either have a G or A, and the rest will be that way. And there are a few right down here in my gene. They're just not in the promoter region. Nonetheless, this is a really nice way to map out your promoter elements, but you can map them out in a way where you can see their relationship both to the gene you're transcribing and the next gene down. So we've used this feature a number of times, and again, it is really nice to be able to not only see it in relationship to the gene models, but in relationship to the data. So that is the sequence search and overlay feature. So I'm going to clear those out for a moment. And now we're going to look at another really large functional piece, which is the genome viewer. I am going to select this model. And I'm going to either, again, you can right-click to access this menu, or you can use the view and access it. And we're going to choose View Genomic Sequence and Sequence Viewer. And I'm going to make it full screen so that you can see it easily. What this is giving us is the actual genomic sequence of the entire model. That is the untranslated, the translated, the introns, and everything. And based on the annotation itself, IGB will mark out the start and the stop with the green and red boxes, and we'll use color to indicate exons in yellow and introns in white. If this color scheme is hard on you, there is a different color scheme which puts blue and black text on white. You can see in this format, we use blue to indicate exons and black to indicate introns. Now, sometimes you want to see just the cDNA, just the transcript. You click Show cDNA, and all of the introns are removed. You can see just the cDNA sequence. When you're in the cDNA sequence, we also, now you can do this from Genomic, but obviously it's a little more practical to use cDNA, but you can see the translation frame. You can choose, for instance, all the plus translations, and here they are. And you can see the +3 in this case starts with methionine and ends with the stop codon. You can even choose to turn off one of the translations. Seeing these is obviously very nice, but being able to use them is even better. So we have a lot of copy functions. For instance, if you copy DNA with lowercase introns and start and stop codons, what that will do is copy the entire genomic DNA sequence, and anywhere that there's an intron or the start and stop, those will be copied out in lowercase so that you can get some of that annotation information. If you copy annotated translation, it will only give you the +1 frame translation that's annotated to this gene. But sometimes you want more. Let's say what I wanted was actually turn off those. I want to see the minus strand translations, and I think something's going on in there. So I'm going to click and drag until I copy the full reading frame. But what I want to see is I want to copy the selected translations. Come over to my text editor. I'm going to paste it out. But see, what IGB does is it saves the entire translation and tells you what frame it's in for each separated. It doesn't try to intermingle them, which is very helpful. Another thing you can do is you can copy the selected DNA sequence, which will only give you the DNA sequence, but especially in an output like this, it's very convenient to know what was the DNA sequence that you used to generate these frame translations. When we get to alignment sequence, there's even more helpful things that we can do. Now, before we actually look at the next round of sequence, I'd like to point out that IGB by default takes all of your RNA-seq reads and puts them in one track, because it generally, depending on the technique you use, doesn't matter the direction it was sequenced, it only matters the position it was aligned. Now, there are stranded type techniques that it does make a difference, but these generally do not. You can, of course, always choose, once again in the annotation, to separate them by the strand they were aligned to. Here's the plus strand and here's the minus strand. But it also does fill up a lot of extra space. I'm going to identify which strand, which reads are on which strand by using this function, color by strand. When I turn it on, I have chosen that all positive strand, that is 5 to 3, are going to be colored green, and all negative strand, the ones that are running 3 to 5, will be colored in gold. The reason I want to show this to you is because if we select, let's say, this read, I'll zoom in a little bit so we can see it highlighted. This read has a gap in it and has a mismatch in it. And we have two options. We can either, once again, view genomic sequence in Sequence Viewer. And what this is showing me, now notice we're on the minus strand. So the little tail is what's listed first. And this longer section is what's listed down here. But what this is actually showing me is the genomic DNA that this maps to. This is not the sequence of the read. I can, again, from the view screen or from the right click, I can view the read sequence in the Sequence Viewer. And now we can actually see the difference. Because the genomic is running in one direction and this is running in the other, I'm going to actually use the reverse complement function. is comparable to the genomic sequence. We can now see that they do run in the same direction. However, when I look at the read sequence, it does not have this included. And right here, if we look, there's two A's right there, CAA. But our read actually does not have CAA. It has CGA. This G in the read should be A according to the genomic data. And we can actually use the genomic sequence viewer to identify that. Another time that that's useful is a read such as this one. What this has in it, this bright red AT, indicates that there's an insertion in this read. And we can select the insertion itself, and it tells us that the letter that was inserted is T. We can select the whole read again, and it will highlight the insertion like this with this black, or if we go back to the original color sequence, it'll show it as a white T to indicate that there is the insertion. That is the various ways you can use the sequence viewer and the sequence overlay in combination with your data analysis to get more information out. Once again, this will be a monthly feature where we'll talk about some element of IGB and go in depth on it. If you'd like to do a specific tutorial, if there's a feature, a function, or a way you'd like to use IGB that you haven't seen yet, you can always contact me and we will set up a private tutorial for you or your lab.